Welcome to the fifth part of the PamGene data analysis training. In the previous videos, we have covered experiment design, image analysis, basic processing, and the analysis of phosphosites and kinases. The outcome of the phosphosite and kinase analysis are significant phosphosites and kinases. How can we interpret these results? Which signaling pathways and networks are playing a role in our model system? This video provides a short guideline into how to interpret PamGene results and walks you through some pathway analysis tools. First, I would like to talk a little bit about the nature of phosphosite and kinase data. PamGene identifies activated kinases in the lysate by detecting phosphorylation of their targets, which are the phosphosites on the PAM chip. Phosphosites have been mapped to proteins by the BLAST tool, and one phosphosite can be found in multiple proteins. However, even though phosphorylation of a certain phosphosite is detected, this does not tell us that the protein is present in the sample. Additionally, it is also good to keep in mind that an increase or decrease in phosphorylation of a phosphosite does not necessarily mean induction or inhibition of the protein. Therefore, we recommend to primarily use kinase predictions to explore signaling pathways. The interpretation of kinase predictions is also more straightforward. Positive fold change means activation and negative means inhibition. However, we have to keep in mind that kinases are not directly observed, only their activity. There is a many-to-many relationship between kinases and their targets, meaning that multiple kinases phosphorylate the same phosphosite and that one kinase can phosphorylate multiple phosphosites. The upstream kinase analysis algorithm deals with this complexity and predicts kinases with two scores, the specificity and the significance score. Therefore, since kinases are predictions, we recommend to use kinases with high scores to explore signaling pathways. Our default threshold is final score 1.3, but you can fine-tune this threshold based on your research question. To explore pathways next to the kinases, we still recommend to use the phosphosite level data as well. This is because at first we just want to get an impression about the signaling pathways working in our model system. So for this initial exploration, it is okay to make these simplifications. If you use pathway analysis tools that require fold change as input, when phosphosite and kinase data overlap, we recommend to use kinase level data. Next, let's dive into the technicalities of pathway analysis based on Paintomics data. As a first step, we recommend to use a simple method for pathway analysis provided by the free Metascape tool. You can find the link of Metascape under the description of this video. Metascape is an overrepresentation-based pathway analysis method. These methods test whether the overlap of pathway members in the data is higher than by chance. They require an input of only significant data. Fold change is not needed. So, let's make this list of significant kinases and proteins. For each comparison, we have to use two kinds of files, the output of the UKA and the output of the MTVC or t-test, and both for PTK and STK. For the phosphosite level data, you need to change the output to include the UniProt IDs and gene names as well. For this, you can refer to video 3 at 13 minutes and 45 seconds. Here is an example project. We filter the UKA files for median final score higher or equal to 1.3. Copy the kinase uniprots from both the PTK and STK and paste them to a new Excel. I like to make a note of where the data is from, so I put that to another column. Next, let's filter the MTVC results for p-value less than 0.05. Because the data is redundant, let's take only the unique proteins with the help of the online Venni tool. You can see that we selected 73 lines. When we paste it to Venni, we get only 50 which are unique. Let's paste this into the Excel file of significant kinases and phosphosites. This should be done for both PTK and STK data to get one list. This can be now input to Metascape. Copy-paste the gene list, submit it, and click Custom Analysis. First, you can see here that the uniprots are converted to entras identifiers. The next section is annotation. This is not pathway analysis yet, just an annotation of genes one by one, for example, with gene symbol, etc. I usually don't need any of this, so I untick them all. The next section is membership. This is similar to annotation. The next section is the enrichment. You can find the parameters of pathway analysis here. You can select your favorite pathway databases. Mine is KEGG, Reactome, and Wikipathways. I recommend to keep quorum protein complexes. I will untick now everything else to focus on the main things. Then you can run enrichment analysis, which usually takes a few minutes. It has finished, so we can go to the analysis report page. First, let's download the zip file. Then let's look at the results. The bar plot of pathways shows you the top pathways with a p-value coming from the Metascape algorithm. Importantly, Metascape doesn't just output loads of pathways, but it has a clever algorithm to show which pathways are non-redundant, meaning pathways that don't have many overlapping members. That is what you can see here. When you use your own data, you will surely notice irrelevant pathways to your model system. This is common in pathway analysis, since a lot of pathways share members. However, with the help of Metascape, we can find the most relevant pathways. Each pathway here is actually a list of similar pathways, and the one with the best p-value is chosen, but this doesn't necessarily mean that it is the most relevant to your model system. So, let's look inside the zip how we can further select the most relevant pathways from these. In the enrichment_go folder, there is a file called final_go. Column X is the group ID. In this file there are 65 groups, meaning 65 non-redundant pathways. Let's look at only group 2. Column Y and Z shows that the first in this group is the pathway called signaling by receptor tyrosine kinases. Well, this doesn't say much, does it? So we can check what other pathway this is redundant with, for example VEGF and EGFR. Now we are one step ahead. In columns T and U you can see the members which are also in your data with entras identifier and gene name. Let's go back to the Metascape results. Figure 3 shows the protein complexes from the quorum database. These are directly interacting proteins in different complexes in your data. If you scroll a bit below, you can see their annotation. Similarly, each complex here is annotated with multiple redundant pathway names. I have shown one way to explore which pathways play a role in your model system in test versus control. But how are they changed? Metascape does not use fold change as an input, therefore, we do not have information on how these pathways changed. Next, I will show you a simple tool to look into this question. Paintomics is a great tool to explore how the members of pathways have changed. This is an example output of Paintomics, the insulin KEGG pathway colored by the fold change of a test condition versus control. Using your domain expertise, this helps to draw your conclusions about what is going on in your model system. Let's see how to get here. You can find the link to the Paintomics website in the description of this video. You can use this website with or without a free account, but I recommend to make one so that your jobs are stored. In the left menu at the resources, you can find tutorials, documentation, and example data to make sure that you are uploading your data in the right format. Importantly, Paintomics uses entras identifiers or gene names and not uniprot identifiers. So you can use gene names or translate the uniprots to entras IDs using the ID mapping tool in Uniprot. You can find the link in the description of this video. Next, you also have to make sure that each gene turns up only once. So for the proteins that turn up multiple times, you need to take the mean. Let me show you how you can do this in person. Here is an MTVC app. At the end of the app, in the Select Export step, you select what you want to export. I have duplicated this step. I added the test condition, the UniProt ID and gene name in the rows, and the log fold change in the y-axis. Since the ID is not in the rows, you can see that there are multiple values for the different IDs. You can try it out to add the ID to the rows, and then you should see that each cell contains only one value. However, the mean operator calculates the mean of all the values in one cell, so this is precisely what we want. There is one more thing to do. You have to set a filter for p-value. From the ANOVA step, bring the p-value treatment factor to the filters and set it. Now you can also see that some IDs are filtered out, and this leads to some genes disappearing, if all their peptides have not been significant. We run this step, and the next step will be the Select Export step, where we add the factors to the rows, which needs to be exported. The output relation of the mean operator is named as value. I named the export step as the mean of significant proteins. Next, we need to make a table with the significant proteins and kinases and their fold change. So let's paste the gene name and log fold change from the significant MTVC and UKA results into one document. The fold change from MTVC is called value. The fold change from UKA is called median kinase statistic. Let's also add in the third column the data source. As I have pointed out before, when peptides and proteins mapped to phosphosites overlap, we recommend to use kinase-level data. So let's highlight the duplicates in Excel and delete those that come from phosphosite data. For Paintomics, we need a table with an ID column and a fold change column, so we have to get rid of the third column and save the file in TXT format. Please be careful to use the normal TXT format, not the Unicode text. Now we can upload this file to Paintomics. First, let's select an organism. I will select human. If you are using another organism, please be aware that PamGene uses human proteins to map to our IDs and human kinase databases. Next, we have to upload our data. The great thing about Paintomics is that you can use multiple data modalities. For example, if you have RNA sequencing data from the same system, you can also upload that. Now we will only upload one file. This can go to the gene expression. Here you can select the pathway databases. I suggest to keep to KEGG since it is much harder to interpret the figures from the Reactome database. You can click on the green Run Paintomics button. Next, you can see the ratio of mapped or unmapped features. Unmapped features are those which are unknown to Paintomics. We have all our features mapped, so we can go to the next one. In the results, first, you can see a pathway classification figure, which shows which larger categories your pathways belong to. If you scroll below, you can see the pathways. You can order the pathways by the number of matching features and open one with the brush icon. The pathway map is interactive. You can click on it to see the annotation of the gene. Another nice feature is the heat map. Select all features and click Apply. Now you can see a fold change heat map of all your input data. I have shown you two tools for pathway analysis. The Metascape tool helps you to arrive to a short list of pathways that might play a role in your model system. Paintomics helps to understand how these pathways have changed. In the next step, you can also break free of the limitations of pathway analysis and dive further into networks. We suggest to explore the networks since pathway databases are limiting in what is annotated in a pathway. Therefore, you might miss less well-known proteins that are still in your data. For network analysis, you can use the online string database. You can find the link in the description of this video. String requires a list of uniprots that you already have. Paste it and run the analysis. The network that you get is usually huge and hard to interpret. So, as a first step, I suggest to use make the interaction data more stringent. In the settings, you can untick some of the interaction sources to keep the best ones. Click on Update and you can see that the view is already clearer a bit. Next, what also helps a lot to cluster your results. The clustering methods require an initial guess of how many clusters you expect. You can play with this setting. I will set it to 10. Based on this, you can maybe already see some bigger pathways emerging. For example, DNA damage and repair proteins that affect the cell cycle are here. You can see that, for example, the CDK proteins connect this cluster to the red, orange, and yellow clusters. When you notice an interesting interacting partner of a protein you think is important, you can also click on the edge between them to see where this interaction was derived from. When you have formed your hypotheses about the signaling, we suggest you to validate the strongest kinase predictions based on these guidelines. You can stop the video to read it, and you can also find these guidelines in the PPT.